Hello, good morning. This is Ibukwan Folami, and today we'll be looking at ATP as a source of energy in animal cells. Some people call it the energy currency in the animal cell, and of course, an essential part of energy production is what we call redox reactions, and we'll be looking at this in detail. How all the redox reactions that we considered in SS1 chemistry, do you remember? Yes, so we'll be looking at how they really play an essential role in energy production and energy metabolism in the animal cell. And we know that mitochondria is where energy is produced. That is very basic knowledge, and we've learned that from earlier courses. And we'll be looking at how everything basically adds up together and how all this happens in the complex structure of the mitochondria in the process of the electron transport chain. Okay, so we look at the details of the mitochondrion. So mitochondrion for singular and mitochondria for plural. So now the structure of the mitochondria is very important in the sense that sometimes I notice that students cannot interpret or translate this three-dimensional structure. They cannot relate or interpret it or translate it into this two-dimensional structure. So this is a three-dimensional structure, and this is a two-dimensional structure. In whatever form you see the mitochondrion, it's important that you are able to interpret it. So think, for example, the outer membrane is a phospholipid bilayer. So meaning many times in a three-dimensional structure, you see it as a double membrane. So double membrane. So that double membrane is what we interpret in a two-dimensional structure as a phospholipid bilayer. We are already used to this in Zoo 111 and some other prerequisite courses that we have taken in the past. But this is how the membrane looks like. In the inner membrane of the mitochondrion is what actually folds into the cristae, as we call it. So it's a folding that helps to increase the surface area of the mitochondrion. And that inner membrane of the mitochondrion, the same way we have the outer membrane as a phospholipid bilayer, like with a double membrane, is the same way the inner membrane is a double membrane. So it's good to understand that this is what it translates to as a three-dimensional structure. And of course, the inner matrix, so what you have there, inside the inner membrane, you have the matrix. And of course, the matrix is the same compound, the same structure, I beg your pardon, that we have here. Okay, so this is very important because depending on what we are talking about, because we are looking at energy production, we will need to understand exactly where we are. So that question of where are you, when we begin to go into the details of glycolysis, where is it happening? When we begin to go into the details of the Krebs cycle or the citric acid cycle, where is it happening? It's very good, very, very important in this course. Okay, so in the next slide, Yeah, so it's always very important to understand o- oxidation and reduction in terms of addition of electrons, removal of hydrogen, or loss of electron, as the case may be. So if you remember in your new school chemistry, Ababio, under your chapter on redox reactions, oxidation is defined as addition of oxygen, removal of hydrogen, or loss of an electron. And in the same way, reduction is described as removal of oxygen, addition of hydrogen, and gain of electron. Sometimes it's not very easy to appreciate how this happens, and trust me, the only way you can do this is really going into the details. But I know for some of you, you're not scared, so I will need to show some structures, because that's the only way I can explain this. So it is important that we consider at least oxidation and reduction under these two headings. So in this case, we will consider oxidation as removal of hydrogen or loss of electron. Okay, it may be easy to first look at how the electrons are moving all over the place before we look at the removal of hydrogen. Okay, so we shall be considering NADH and NAD plus as a case study, because this compound NADH, nicotinamide, adenine dinucleotide, it is a compound that is regarded as a high electron carrier. So NADH is regarded as a high electron carrier, and in the electron transport chain, which we'll see later, we'll see how it's involved in transporting electrons from one membrane protein to the other. But in this case, when we are talking about NADH as electron carriers, we are looking at series of redox reactions. So in this case, NADH usually at the beginning is in a reduced state. So take for instance NADH in this form highlighted, it's in a reduced state, and we call it reduced NAD plus. However, in this form highlighted, NAD plus is in the oxidized state. So we'll be looking at what exactly happens during the transfer of electrons that will make us understand how oxidation and reduction is taking place. So keep in mind, we're looking at oxidation from the perspective of loss of electron and removal of hydrogen. We'll tackle that first. So NADH here loses electrons along with this hydrogen and becomes oxidized to NAD plus. And as you can see, the reaction is a forward and backward reaction, so it's reversible. So if you consider NAD plus in the oxidized state, what happens to it? It gains electron and becomes reduced to NADH. I take that again. NAD plus here gains electrons and becomes reduced to NADH. Okay, meanwhile, NADH loses its electrons as well as hydrogen and becomes oxidized to NAD plus. With some equations, you may be able to see exactly what happens. So, take for example, this is a more detailed structure of NADH and NAD plus. Now, we can see exactly how NADH loses this hydrogen when converted to NAD plus, and why we say that in the process it is oxidized. So during oxidation from NADH to NAD plus, the hydrogen here is lost, as you can see here, and it becomes oxidized to NAD plus. But if you're going by the backward reaction, hydrogen here is added to NAD plus in the form of hydride ion. So in the form of a hydride ion, hydrogen is added to NAD plus to become NADH. Addition of hydrogen, reduction. So we can say that NAD plus is reduced to NADH. Meanwhile, NADH is oxidized to NAD plus. So in the process of reduction, hydrogen is added in the form of an hydride, and in the process of oxidation, hydrogen is lost. Okay, so how do we see what is happening with the electrons? With this, I mean, it's obvious how hydrogen is added in one and removed in the other, but what happens in the case of electrons? Okay, now we understand oxidation as loss of electron. So how will NADH here lose its electrons to become NAD plus? So the equation to look at in this structure is the forward reaction. So everything begins from hydrogen. So if you look at this hydrogen, hydrogen here is lost because in the process of losing hydrogen, the electron that form this covalent bond is lost. So 2 electrons are lost here. Meanwhile, to cater for the loss of these electrons, there are electron shifts from the bonds, from this bond to that bond, and in the process the lone pair on the nitrogen is transferred to this bond, and we can see how the NAD plus is formed through the loss of these 2 electrons. And these 2 electrons is precisely what is accounted for here. Meanwhile, the hydrogen is lost in the form of a proton, and we'll be seeing the fate of this proton when we're considering the electron transport chain. Okay, so the next thing is how energy is produced in the animal cell. So we know that we produce energy from the foods. So our foods serve as cells. So when we take carbohydrates, you recall in NAT 201, your carbohydrates are broken down to glucose, your proteins are broken down to amino acids, and your fats are broken down to fatty acids. But these 3 are the fuels that are used to generate energy. So for carbohydrates, essentially, everything boils down to glucose, and energy is generated from glucose. For proteins, everything boils down to amino acids, and energy is generated from there. And for fat, everything is broken down to fatty acids, and energy is generated from there. So it's good to know that these compounds are largely smaller. They are way smaller compared to their macromolecules. We did this in NUT 201. When you hydrolyze, you form your glucose, and when you remove water, you form your carbohydrates. So we did this last. So you remember the glycosidic bonds, the formation of glycosidic bonds. It happens through a process of dehydration, and of course the reverse reaction happens through the process of hydrolysis. So it's good to also know that majority of the ATP production takes place in the mitochondria, and but energy can also be produced outside the mitochondria.